Okay, so our next contestant is Gaurav Agrawal, who's a bioengineering student in the lab of Professor Shiny Varghese. So welcome to Grad Slam.
Thank you. Cancer has become a global epidemic. Did you know that it's expected now that 1 in every 3 Americans will develop cancer at some point in our lifetimes? And globally, the numbers are worse. And since we just don't know when or where cancer is going to develop in our bodies, as scientists, I believe we must focus our energies on developing effective medications to fight the diseases that we know are coming. Unfortunately, cancer drug development is no simple task. Pharmaceutical companies currently spend 10 to 15 years and upwards of $3 billion developing and testing a single cancer drug. And despite this immense investment, over 90% of drugs still end up failing before ever reaching patients. So you might be puzzled. Why is this failure rate so high? Well, in large part, it has to do with the current drug testing methods themselves, which are inadequate. These include testing drugs on cells in a plastic dish or in animals, such as mice. Now, do either of these two mimic a human to you in any way? No, of course not. And it's no wonder that so many drugs that pass these first 2 stages ultimately fail in humans. What if there was a drug testing system that better mimicked the complexity of the human body? Well, in my research, I have focused on developing such a platform, and I think it's going to help us find some promising new medications. I have developed a device that houses small pieces of liver, heart, and cancer tissues made entirely from human cells. And when you link these 3 individual devices together into one continuous system, we call this the human body on a chip system. In this system, tissues can actually communicate with one another through a fluidic circuit that we have built that mimics the blood vessel network in our bodies that connects all the organs and tissues of our own bodies. And with this dynamic system, we can look at not only how effective a drug is against the cancer by looking at how much the cancer might grow or shrink, but we can also look at the damaging side effects of the cancer drugs. For example, irregular or abnormal heartbeats. Moreover, we've actually been able to demonstrate with our system some clinically accurate results. In fact, using a common cancer drug that is known to only work once it's been metabolized and activated by the liver, we have shown that when that drug is tested on cancer cells in a dish, the drug fails to work and the cancer continues to grow. However, when you test that exact same drug in the human body on chip system that we have built, the drug indeed gets metabolized and activated before proceeding on to destroy the cancer altogether. This is the correct response as seen in human patients today who take this drug. And this highlights the importance of the complete body-on-chip approach, which is missing entirely from the Petri dish model. Moreover, as compared to animals, our system is about 50 times cheaper than a standard mouse, and it takes just days to develop as compared to weeks and weeks of breeding and nurturing mice. So we hope that our system will be able to revolutionize cancer drug development by reducing our need and reliance on animals. Ultimately, my goal is to help democratize drug screening in order to expedite the discovery of cancer drugs and quickly find the cures that we need for a brighter and healthier future. Thank you.